هنبدأ السيشن 6 workshop The Microbiota Revolution، ويسعدني ان انا اقدم one of the eminent speakers and our eminent scientist in Egypt دكتور استاذي ومعلمي دكتور نادر فصيح هيكلمنا The Microbiota and the Lung. اتفضلي.
بسم الله الرحمن الرحيم. طبعا أنا يسعدني ويشرفني كلمة الأستاذية بما تعنيه الكلمة من معنى أستاذتنا الأستاذة الدكتورة سناء وجميع التيم اللي معاها، سواء دكتور إيهاب، دكتورة مروة، دكتورة ياسمين، دكتورة مي، المجموعة كاملة حقيقي بيقدموا أوركسترا عظيم بيعزف أجمل الحاجات اللي بنقدر نستمتع بيها ونستفيد منها كلنا. طبعا دكتورة تريز على راسي من فوق، هي مش يعني هتعجزني قوي، لكن هي طبعا هي حبيبتي وبنتي واختي وزميلتي وصديقتي، يعني ربنا يكرمها ويبارك فيها. الساده الافاضل الدكتور ماهر، دكتوره مروه، انا سعيد جدا. هتكلم بسرعه على موضوع الدكتوره سناء امرتني ان انا اتكلم بيه، وبالتالي ما ينفعش هي تؤمر وانا ما نفذش. The objective of these presentations first to take some definitions which are very important about definitely what is microbiome, what is the microbiota, the colonization, the carrier state, the infection state, and the infectious diseases. We should discriminate between all of these definitions. Second is that the respiratory tract is a complex organ system which reveals a broader role of microbiota in the human health. The third point is that definitely recently we understand through the next generation sequencing اللي هو reveals advances with understanding of the microbiota of the lung, which is not yet well proved قبل كده. كلنا عارفين اللعبة دي ولعبناها زمان واحنا صغيرين، عارفينها طبعا ولا نسيتوها؟ ولسه بيلعبوها ولا ما بيلعبوهاش؟ عارفين لعبة الكراسي الموسيقية طبعا. لعبة الكراسي الموسيقية احنا عندنا عدد من الكراسي أقل من العدد اللي بيدوروا، وكانت لعبة جميلة جدا ولطيفة جدا وبنحبها خالص، لكن كان في واحد بيطلع لأن عدد الكراسي أقل من العدد اللي بيدوروا. This is the same concept for colonization resistance, which is of paramount importance of respiratory health. Definitely what is known as colonization resistance. This is a very important term which should understand it well. The colonization resistance is the basics of understanding how the microbiota of the respiratory system protect against diseases. How it comes this? If we have healthy microbiota in the respiratory system, yes, this healthy microbiota, it has a very big and important role. Through what? It will prevent the pathogenic bacteria from growing inside. How it comes through that? We have some bacteria or some healthy bacteria which is very important for consuming all the nutrients instead of the pathogenic bacteria. This is number one. Or through secretions of a lot of cytokines, chemokines, and the chemical compounds which prevent even the biofilm or the multiplications of the others or preventing adherence of others. We have different definitions like what is meant by microbiome. Do you know what is actually meant by microbiome? Microbiome is all the genetic contents of all the microbial community inside our body. Yes, this is all the genetic contents. And what is definitely meant by microbiota is the microorganism, including all microorganisms, whether this is bacteria, virus, single cell lived organism inside our bodies, inside each particular niche inside our body. We have different sites in which we have microbiota, which is commonly known as the skin microbiota, the vaginal or genitourinary microbiota, intestinal microbiota, and respiratory microbiota, and these are the common microbiota inside our body. What's actually meant by colonization? Colonization is the presence of the microorganism in the host. Yes, and what else? It doesn't, and this organism can be multiplied, but there is no interaction between this microorganism and the host. So this is very important for determining colonization. But we have a very big problem. Can you differentiate between a carrier state and a person with colonized bacteria or colonized viral? It is so much difficult to differentiate between the carrier state and the colonization. Between the carrier state, it can give many possibilities that will be described in the next slide, yes. Infection, here we have an organism that interacts within the body, whether this will produce a disease state or not. And the disease state, this is the results of the interaction between the organism and the body, which results in definitely a disease state. So the carrier state, if you take a throat swab, for example, and you will get some bacteria, for example, streptococci, for example, whatever the species of the streptococcal infection that you get it. This means that if this patient is colonized, or this patient is definitely infected, but in an asymptomatic state, or this patient in the incubation period before the presence of the manifestation of the disease, or this patient definitely in a stage of convalescence from the state of acute disease. So the term carrier state is a very complex term, which means a lot of issues. One of these is the colonization state. Definitely infection is an interaction between the organism and the body with or without the presence of disease itself. Definitely, are we born sterile? Definitely no. We may get recently some of the organism in utero, and this will be definitely later on. But for the respiratory microbiota first, definitely which depends on two main issues. First is the birth type, whether cesarean section or vaginal delivery, and the second also is the second important issues is the feeding pattern of that baby. So we get the nasal or nasopharyngeal or oral microbiota of a newborn at day 1 or 0 time at 1 hour after delivery. We may get different types of bacteria which is never known to us. But if we pass to the first week, we get multiplications of staphylococcal infection in the nasopharynx, nasal, nasopharyngeal, and oropharyngeal site of the upper airway. And then we get another two important bacteria, which is Corynebacterium and also Dolo-Segrenalum bacteria, which is very important. These two types of organisms are very important to be present early in the first 3 or 4 months of life because they have a very protective effect against the colonization of many pathogenic organisms. And then we get later on in life, at the start of the 4 months or even 6 months, the colonization with Moraxella catarrhalis. And so it is very important to know that the time and the type, two issues, which type of bacteria that will be implanted in the upper airway first, then the different types, and then the timing of implantation, which are very important for making the respiratory system is very healthy. This is an example of the Keystone species of the organism, and then how there is a state of symbiosis between the different types of the bacteria. The term symbiosis means the interaction between two different types of bacteria and how it can affect the growth and the colonization of the other bacteria. We have two examples that we can say here, the Dolo-Segrenalum bacteria and the Corynebacterium. These are very important for preventing and strongly associated with the respiratory health, especially in protecting against Streptococcus pneumoniae. So early implantation and the colonization with these types of organisms in the first few months are associated with protection against Streptococcus pneumoniae. Also, Staphylococcus epidermis is very important in that they prevent the biofilm through the Staph aureus infections. Through what? Through the productions and secretions of serine proteases, which are very important for preventing staph biofilm productions. We know, all of us, that we have many types of microbiota, like upper GIT microbiota, and I think that Dr. Khairi, Dr. Sana, Dr. Yasmin will discuss this in details later on, whether the upper intestinal and the lower intestinal, and how they play a major role for our health, and how they have an interactions between the other immune status for other organs. Also the conjunctiva, the skin, for example. Also, we have different types of skin flora, and this is very important. And you can say, in the next slide, how we have different even microbiota in the different types of skin. And then the respiratory system, we have different types of microbiota, whether they are in the nasopharynx, oropharynx, and even the lower lung. We should know that the human lung شقة مساحتها 70-130 متر تقريبا، وبعدين عندنا 100,000 اير واي، عندنا 200 مليون alveoli. البادي contains about 10 raised 3 multiplied by 10 raised to the power 13 cells inside our body. But we have more and more bacteria inside our body, which is 3.8 raised to the multiplied by 10 raised to the power 13 in our body. So we have more microbiota inside our body, more than the total number of cells. لو بصينا على عالم قطر والبحرين هنلاقي فيه تشابه كتير جدا. اللي يفرق بينهم اللون الأحمر أغمق شوية، المسافة أقل شوية بين الأحمر والأبيض، وبالتالي الاختلاف بسيط جدا. The same as we can see here how in the skin microbiota we have different microbiota between the dry, moist, spacious glands and also the food. So we can get different types of microbiota which have a special role for preventing infections in each site of the body, which depends on a lot of issues that we can say here. Childhood asthma risk factors is very important for all of us. We should know that genetic risk factors nowadays, we know that the genome-wide association studies, the genome-wide association studies definitely resolve a lot of problems that we are not yet understanding. And how we know that we have many genes like orozomycoid or Gisterman genes, which are very essential. They are present on chromosome number 17, and which are related to definitely allergic asthma and type 2 inflammatory asthma. Also, we have a lot of genes which are well known to us to be present, and they are definitely related to the development of allergic manifestations. The environmental risk factors like maternal tobacco smoke now is associated with a very high risk of development of asthma. Maternal diet, for example, polyunsaturated fatty acids. Polyunsaturated fatty acids, vitamin E and zinc are associated with low risk of asthma developments. In contrast, any high sugar content during pregnancy definitely will be associated with an increased risk of asthma developments. Still, the Mediterranean type of food, vitamin A and D, are still controversial for its role in preventing asthma developments or not. Caesarean section definitely has a high association with the asthma developments. However, this may be due to the affection of the flora of the respiratory tract that may affect definitely the progress and respiratory health and development of asthma. Natal risk factors like chronic lung disease or prematurity, caesarean section, again, extreme preterm babies and low birth weight babies definitely are associated with increased risk of asthma developments. Even boys are more risky to develop asthma below the age of 15, but beyond that age, a little bit, girls are more associated with asthma developments rather than the boys. The Positive family history of atopy, especially in the mothers, is associated definitely with increased asthma developments rather than the father, and the atopic march and the early sensitization with food is associated or are associated with asthma developments more in early childhood. This slide is very important to all of us to know how we have different sites in the respiratory system with different microbiota, and why we have different microbiota at different sites. We can say here that according to the pH, the carbon dioxide tension, the oxygen saturation, that will affect definitely the type of the organism. All of us, we know that we have aerobic or anaerobic, or which types of organism which you prefer, high oxygen content or low oxygen content. And also we can say how the surface area that the microbiota will stay. We can see how much the microbiota in the lower lung is about how many times that in the upper airway. It is a huge space for microbiota if compared to the oropharynx, and that the nasal cavity contains about 60 times or 70 times more than the microbiota, or 10 times or 15 times the microbiota of the oropharynx or the nasopharynx. And we have different types of organism at each site, which are completely different from the other types. So the microbiota of the nasal cavity is rich in staphylococcal infections, then Corynebacterium, Moraxella, Streptococci, and then if we pass to the nasopharynx, we get Moraxella, Staph, and the two important organisms, which is Corynebacterium species and Dolosegrarum, which are very important and protective against the presence of invasive and pathologic Streptococcus pneumoniae. And also how in the lung and also in the trachea, the presence of Veillonella. Veillonella is a very important bacteria which help what? Help the infection and the colonization with the streptococcal infections. So the presence of each type of these bacteria in the NIC or NICE is very important to justify which type of organism that you are liable to develop and which type of infection that you are liable to get. But as we can see here, how the host and the environment factors that influence definitely the respiratory microbiota, and how the type of birth, whether it is definitely cesarean section or vaginal delivery, each of which have an effect on the microbiota inside our body and protection against infections. Then we pass to the feeding type. I think that Professor Dr. Khairi, Professor Dr. Yasmin, Dr. Sanaa discuss a lot of how the presence of certain types of bacteria and secretory IgA and secretory antibodies that is present in the breast milk, how much they can prevent against the development of a lot of respiratory diseases. The presence of other siblings in the home, definitely they have a major role for the development of respiratory tract infections. أكيد طبعا كلكم شايفين أول بيبي بيبقى كويس قوي، لو 2 أو 3 أو 4 يبتدي الأرقام تزيد جدا جدا، لأن الريسك of contamination infections بيبقى أعلى. طبعا لو بيروح day care centers, respiratory infections is about 3 to 4 times more than if he is in home. السيزون طبعا كلنا داخلين في السيزون بتاعنا 10 لحد 5, definitely we have a high risk of infections. Genetic predispositions, as we said before, the genome association-wide studies definitely signifies which type of genetic susceptibility that they are present, and if they are present, definitely they lead to increased risk of infections. Vaccination also can help much for prevention of other diseases. Infections early in life and the introduction of antibiotic is still a major role for disrupting the microbiota inside our body, and they play a very important role for disrupting the healthy microbiota of our body, especially beyond the age of 4 years. Smoking is very important for preventing for disrupting also the microbiota because it disrupted the mucociliary clearance through stimulation of the goblet cells and disruption of the functions of the cilia, and will help much for the adherence of many bacteria for the respiratory epithelial cells. Lastly, the take-home message that the human respiratory tract spans from the nostrils down to the lung alveoli, and the microbiota of the respiratory tract probably acts as a gatekeeper that provides resistance to colonization by respiratory pathogens. Lastly, the respiratory microbiota might also be involved in the maturation and maintenance of homeostasis of respiratory physiology and immunity. And the last point is that the ecological and environmental factors that direct the development of microbial communities in the respiratory tract and how these communities affect the respiratory health are the focus of current research. And thank you.
ثانك يو بروفيسور دكتور نادر فصيح. الحقيقه كما اعتدنا من حضرتك المحاضره رائعه واكثر من رائعه، وفيها كل مره بنتعلم اسامي بكتيريا جديده.